Right, so welcome everyone. This first presentation is going to be a little bit on the larger side, so I might split it up into a few chunks. But really, the core of this sort of section is to understand the molecular basis of how allergic inflammation occurs. And just because the word allergic is there doesn't mean this only has to do with food allergies. This is going to be relevant to anything really under the umbrella of atopic conditions or these hypersensitivity reactions, and allergies. Asthma are big ones, but also things like eczema, allergic rhinitis, which is more like the seasonal allergies, and even things like psoriasis, mastocytosis, a lot of dermatological conditions as well. So yeah, let's just get into it. This is going to be going over some of the key players that sort of control these molecular inflammation pathways. So really the learning objectives today are to first get into the cytokines and other inflammatory mediators. Some of them are different molecules, but really the things that get released that cause inflammation is, I guess, how I would say that. And then we also want to look at different immune cells, especially these lymphoid and myeloid cells, that have a pretty defined role in allergic inflammation specifically. And then we're also going to go over some certain areas in the body and some certain pathways that are of particular note. So here is the lecture content, so you can get a rough idea of where things are if you want to skip ahead. All right, and so here, if you've taken an immunology course or anything like this, you might have seen a graph very similar to this, but it basically shows how you get from a hematopoietic stem cell, or the HSC, and you get into a variety of these specific downstream cells. So the ones we're really going to be interested in are, first of all, helper T cells and mast cells. And then you also have some other ones that are really important, like eosinophils and plasma cells, which are going to be secreting those antibodies, especially those IgE antibodies. And then these monocytes, like the dendritic cells and macrophages, are also important, but we'll get to these in a bit. All right, so let's just talk about mast cells. And again, a lot of the things that I'm mentioning here are going to have their entire own presentation. So I have a whole thing coming up on mast cells. So we'll just go over a pretty surface level thing. But these are really involved in an early response to allergens that enter the airway, the skin. By early response, we mean an allergic reaction that's happening really within minutes. So if you think of like an anaphylactic reaction, well, these are pretty big players behind that. So these are activated, and yes, they do have to be activated through IgE-dependent mechanisms. And again, we'll sort of get into the nitty-gritty of that later on, but for now, I just want you to know that the mast cells, they're not, typically they're not always just shooting out inflammatory mediators. They have to be activated, and IgE, as I said, is one of the big ways that happens. And when they are activated, all of these chemical mediators are released into the bloodstream. And these are going to cause a lot of the symptoms we see in allergic diseases, especially fatal allergic diseases such as bronchoconstriction, which is tightening of the airway, vasodilation, which is when your blood vessels widen and your blood pressure is going to drop, and plasma exudation, where this fluid begins to leak and draw in more immune cells. So mast cells are, they're found in the airway smooth muscle, among many, many other places. But in the airway smooth muscle, they contribute to something called airway hyperresponsiveness, which these are relatively important abbreviations. And airway hyperresponsiveness is basically when a person's airway is sort of primed to overreact to something. And that's really the basis of a lot of inflammatory diseases in the first place, is that things are just overreacting when they shouldn't be overreacting. And so there are sort of two ways that, or two molecules that are found inside mast cells that contribute to this, among many others. I will mention chymase, which is, not as much as known about it in allergic diseases, but I just want to throw it out here so it's familiar to you in case you see mast cells that are phenotyped by the protease they have inside. So some of them might be tryptase positive, chymase positive, etc. But really the thing we want to focus on is this tryptase molecule. So tryptase is a serine protease, I believe, and it's very pro-inflammatory. It's released exclusively from mast cells, and it contributes to this airway hyperresponsiveness in asthma. So again, a lot of this lecture is not going to be going into the complete deepest dive of any of these things. I'm more so just I want to establish familiarity with them, so that when I bring them up later on, you can sort of recognize where they are. So tryptase, think mast cell, think pro-inflammatory. Yeah. So macrophages. Macrophages aren't something we typically think of being as involved with in asthma or allergies, but they are. And so these can be activated by allergens because they actually carry a low affinity IgE receptor. And most of the time we're going to be talking about a high affinity IgE receptor, but I also want you to know that there's a low affinity one, and we're going to be talking about this again in a different lecture on IgE receptors specifically. But when they are activated by this receptor and interface with an allergen, they're going to cause traffic, cellular traffic into the airways during asthma. So they're going to recruit potentially things like neutrophils or other mast cells or other macrophages, and these are going to all contribute to more airway hyperresponsiveness. And also just a thing to note that there's M1-like macrophages and M2, and the former are pro-inflammatory generally, and the latter are anti-inflammatory generally. So macrophages, depending on where they're found in the body, are going to be called different things. For example, in the skin, they'd be called Langerhans cells, I believe. But in the airways, they're called alveolar macrophages. So these alveolar macrophages are normally meant to suppress lymphocyte function. So lymphocytes thinking like B and T cells. But this can sort of be impaired in asthma, so they might not be doing their suppressive role as well during asthma. And this can lead to more hyperreactivity, right? Because if you're not properly suppressing your immune cells, those immune cells are going to run rampant. Another thing they do is they are going to generally inhibit IL-5 secretion from T cells, and this is another function that might be impaired in asthma. It's a little tricky to think of how to sort of teach this, because again, like, I haven't gotten to IL-5 yet, but IL-5 is very related to cells, so it's worth talking about the cells first. But keep this IL-5 in mind. It's really, really important. And you can generally, if we approach this in a pretty reductive manner, you can think of IL-5 as being pretty correlated with inflammation in general, and it's going to be tied to eosinophils specifically. All right, so dendritic cells, this is sort of a refresher from general immunology, but dendritic cells are really important because they are one of the main ways that we sort of initiate this whole inflammatory cascade in the first place. Because when an allergen enters the body, it has to first sort of interface with our cells. It has to be presented to our cells. Otherwise, the cells are not going to know that it's there, and they're not going to overreact. So these dendritic cells are really valuable because they take up these allergens and they basically cut them up into these peptides, transport them to lymph nodes or other sites where a lot of immune cells are, and they show these peptides, which are allergenic, to naive T cells. And they're, if you've taken an immunology course, you know that there are a lot of co-stimulatory signals that are involved in T cell activation. But basically, the big picture here is that dendritic cells are sort of our link between an allergen entering our bloodstream or entering our body and being actually presented to a T cell, and that T cell becoming sensitized and able to react to it. So here's another really big thing that I want to bring up, which is this idea of a Th2 cell. So that stands for T helper 2 cell. Again, I'll get into this a little bit more later, but Th2 cells are one of the biggest drivers in all allergic diseases. So these are a really, really important thing to know. And specifically, we want to think about Th2 cells in regards to being in a constant balance or tug of war with the Th1 cells, T helper 1 cells. And when there's more Th2 cells, an individual generally is going to be more predisposed to atopic conditions. So this whole process of dendritic cells sort of presenting and polarizing naive T cells into these Th2 cells is really critical. So talking about eosinophils, these are coordinated by these Th2 cells. So now we can see how these Th2 cells are not just sort of in that tug of war, but specifically they are very involved with a lot of these other inflammatory cells. So Th2 cells can release IL-15 or IL-5, so interleukin 5. And this IL-5, as I'll get into later, is really important to the growth of eosinophils. So eosinophils also release some other molecules. Cysteine, leukotrienes are also important. These are big in asthma. And I believe montelukast, which is a common asthma drug, works through sort of inhibiting these types of molecules. I could be wrong. And they also have these cationic proteins that are used in helminth destruction. So this sort of starts a bigger theme that I don't really have time to get into now, but is something that will come up again and again, is why do we have these bad cells? Like, why are they still around in our body? And, where I can, I try to highlight that some of these cells do have important evolutionary functions, like helminth destruction, which could have proved to be something that would have conferred fitness during human evolution. But now we know that there's a lot, much darker side to these cells that contribute to these allergic diseases as well. So just to go over some big, broad overview of lymphocytes, again, these Th2 cells are really important because they're releasing IL-4, IL-5, IL-9, IL-13. Generally, I would say these are considered to be, I guess the main Th2 cytokines that are involved in allergy. And so if you ever see type 2 cytokines or Th2 cytokines, generally they're talking about these ones, and they have very related functions. As I said, B cells, or I guess more accurately, plasma cells, are going to be producing IgE under the influence of IL-4 and IL-13, which come from these Th2 cells. So you can kind of see how all of this is sort of related, how IgE is going to be produced under these, the stimulation of various cytokines, and those cytokines are going to come from, an allergy I want to get across here. And I talked about airway smooth muscle a little bit, but the cells themselves are quite important in asthma. So again, a lot of these mediators that are released from mast cells are going to be responsible for driving that bronchoconstriction that's central to asthma. And airway smooth muscle, that ASM, oftentimes you will see that the mass of that muscle is going to increase quite dramatically during asthma, and this is something called hyperplasia. So again, we'll get into this a little bit later, but just throwing out terms so that we're familiar. And blood vessels are also really important in the sort of function of everything, because this is how things get around, right? So this is not only how a dendritic cell might get around to present or use the lymphatic passages, but cells that when they migrate to sites of inflammation are going to come through these blood vessels. So vasodilation is really important because this is not only a fatal consequence, because if you've taken like physics or anything, you'll know that when the diameter of this blood vessel dilates, the blood pressure inside is going to drop, and when the blood pressure inside drops, leads to a whole cascade of things that could be potentially fatal, especially shock, anaphylactic shock. But it also increases the blood flow to take more of these inflammatory cells around your body, and it causes leakage and swelling. That's where we get this wheel and flare reaction as well. So this vasodilation is generally pretty responsive to histamine as its main signal, but also things like PGD2. PGD is going to stand for prostaglandin D, so this is prostaglandin D2, cystinyl leukotrienes, as we talked about, and nitric oxide, which is one of the big vasodilating factors in the body. So now we can also talk about mucus hypersecretion. So this is something, again, that's going to be really important in asthma. But mucus hypersecretion is driven by these goblet cells that are found in the epithelium and also in our mucosal surfaces. These goblet cells are going to be really responsive to these inflammatory mediators, which is going to basically signal them to grow and grow and grow. That's going to result in that hyperplasia we were talking about. And that is, very linked again to all of these type 2 cytokine secretions. This is something that I'll bring up because I thought it was interesting, but ATP, which we typically think of as a sort of energy force in our body, is very strongly, a stimulator of mucus hypersecretion, and it acts on these receptors. So it's just interesting to think about, how our body might be able to discriminate between ATP as a metabolic factor and ATP as an inflammatory signal as well. All right, so now we're going to talk about the sort of nitty-gritty, and this part can be a little overwhelming because there's just so much to cover. So again, the purpose here is not to completely understand every single molecule or mediator that gets brought up, but rather to develop a familiarity with it, so that when it comes up later on in more specific contexts, that we can sort of recognize the general function. So inflammatory mediators, we can basically say that this is a general term for all of the molecules which are causing the effects of allergic diseases. So if we think of things like bronchoconstriction, or contracting airway muscle, microvascular leakage, mucus production, chemotaxis, all of these things, these are going to be caused by this broad term of inflammatory mediators. And down here you can see a little good overview of how we get from these sort of cells to the mediators, and then the effects. So the mediators are literally mediating the cells and their effects. So lipid mediators are one of the big classes. So we have things like cysteinoleucotrienes. These are bronchoconstrictors generally, and you can think of these mostly in the context of asthma, at least within the scope of this class. We also have leukotrienes, so you would see these abbreviated as LT, so in this case LTB4 for leukotriene B4. So this one specifically is another asthma exacerbator. It activates T cells, and we also have prostaglandins, so PGD2. So this one is a big mast cell mediator, so if you see this, oftentimes it'll be coming from mast cells in the context of allergic disease, and again, this is a bronchoconstrictor. Now, this is a bit of a can of worms, but I'll mention it here and we won't go too deep into it. But arachidonic acid, so this is deeply tied to our, sort of inflammatory pathways. And these are oxidized into F2 isoprostanes. For now, again, what's important is that if you see either of these forms of arachidonic acid, that we just sort of think back to big picture bronchoconstriction, plasma leakage, and that sort of thing. And again, if you're unfamiliar with lipid structure, you can see here long chains, hydrocarbon chains. So again, nitric oxide, I talked about this a little bit earlier, but this is one of the major vasodilators in our body. It's produced by nitric oxide synthase, especially in the airway. And FeNO tests, if you've ever heard of these, stands for fractional exhaled nitric oxide. And these can be used to help diagnose asthma or do other pulmonological tests. And we basically see that these are higher in patients with asthma. So purines, so again, this goes back to that little fragment I mentioned about ATP, but purines, especially adenosine, can lead to bronchoconstriction. So again, this is taking them out of contexts which you might have heard about them more commonly in biology, such as a metabolic context or even a genetic context. But yeah, these are inflammatory mediators as well. And adenosine can react with mast cells. This is one of the many ways that mast cells can sort of receive stimulatory signals. And ATP can also be a sort of danger signal that gestures to tissue damage, and therefore this is going to enhance chemotaxis and activate all of these inflammatory cells that are going to come migrate to that spot of tissue damage. Another thing I want to mention is that we always think of, or at least I always thought of, inflammation as being a very, chemical process. But, not to say it isn't, but the point of this slide is that I also want to bring up the fact that the nervous system is involved in this as well. So when we do things like cough, or we scratch, or we sneeze, these are symptoms that are produced by the nervous system, right? It's not purely chemicals by themselves. So these nerves that are causing us to do these sort of like little tics or coughing, these are activated by inflammatory mediators as well. And so we have things like histamine, protons, which is a correlate for acidity, bradykinins. So all of these things can sort of sensitize these nerves and decrease their threshold of reactivity and cause them to sort of hyperreact. And that is a sort of broad trend with a lot of hypersensitivity reactions. And here, this image over here, so you can see this TRPA1. That stands for transient potential receptor A1. And this is an ion channel that is used in nociception as a sensation of pain. This is elevated during asthma. And basically, you can see that this cycle here is that, these neuropeptides are released, activating on inflammatory cells. These inflammatory cells are going to release inflammatory mediators, which act on the sensory nerves to, cause us to scratch, itch, cough, all of those things. So the cycle is really what I want to get hammered home here. Okay, so now let's talk about transcription factors. So transcription factors are pretty important because, again, it's not just things happening in the blood, it's things happening within the cell as well. So when we say that there are increases in inflammatory proteins, inflammatory molecules, mediators, these are linked directly and they come from these transcription factors, which are telling the cells to make more of them. So one example I'll bring up, and we'll see this one commonly, but there are many, is nuclear factor kappa B, or NFKB. So this is activated in asthmatic airways and regulates a lot of genes that are directly involved with this cellular information. So it controls genes for various cytokines that we talked about, various chemokines, adhesion molecules, inflammatory enzymes. This is sort of a big picture overview and we'll get to this later when we start really getting into pathways, is that, things don't happen in isolation. It's not, all the time, it's not like cells are just storing cytokines and are ready to release them. Sometimes that is the case, especially with mast cells. But we really want to think about how that pathway occurs all the way down to the level of the nucleus. And we're getting these transcription factors involved as well. So this is another transcription factor that I'll talk about, and this is GATA3. As you can tell from the title, this is going to be pretty important because this is a transcription factor which really polarizes uncommitted T cells and urges them to sort of become Th2 cells. And we know that if we have more Th2 cells, we're going to have more, allergic inflammation. So we see that, unsurprisingly, in asthmatic individuals, we have way more T cells that are positive for GATA3. But we also see that it can be downregulated by cytokines such as IL-27. And this is going to also be a theme where I'll sort of bring in some treatments that are commonly used. In this case, corticosteroids, such as, for example, cortisone. And I'll talk about a little bit how they work. So one of the ways that corticosteroids work to help, for example, eczema, is that they're going to rapidly inhibit this transcription factor. And the way it does that is it competes for a nuclear import receptor or protein. Obviously competition at the level of the nucleus is going to interfere with these transcription factors. And another thing I want to talk about is that it's constantly in balance with its sort of other transcription factor, which is TBET. So if I talked about earlier how, for example, Th2 cells were in conflict with Th1 cells as a sort of constant balance, here we can think of one level above that is the GATA3 transcription factor is in a constant balance with the TBET transcription factor. So here's a pathway I want to talk about, which is the JAK-STAT pathway. So JAK stands for Janus activated tyrosine kinase, and STAT stands for signal transducer and activator of transcription. So basically, big way of saying that these are, a sort of transcription factor that are regulated by these kinases. So together, it's a JAK-STAT pathway. So this is going to be sort of another level of regulation on top of the Th1 versus Th2, on top of the GATA3 TBET. So depending on the cytokine that stimulates the tyrosine kinase, we are going to get a different result. So if you have IL-4 and IL-13, here's a little quiz moment. Hopefully we can remember that these are generally type 2 allergic cytokines. So if we have these allergic cytokines and they're stimulating these JAK1 and JAK3, this is going to lead to, again, this is just a big downstream pathway, but phosphorylation of STAT6, activation of that GATA3, and finally arriving at that Th2 differentiation. Conversely, if we have IL-2, for example, IL-2 is going to activate different kinases. So we're going to activate JAK2, tyrosine kinase 2. This is going to result in phosphorylation of not STAT6, but STAT4. And this is going to activate the TBET transcription factor. So again, there's just so many things that regulate this Th1 and Th2 balance that I want to sort of bring to our attention. We've talked about the cells, we've talked about the transcription factors being GATA3 and TBET, but we also have these other transcription factors, STAT4 and STAT6, and we also have these cytokines, and we also have the kinases that are acting on the cytokines. And again, it's not the most important thing to remember which, these entire pathways, but hopefully if you see something like GATA3, that could be setting off a little signal in your brain to start thinking about Th2 cells, for example. All right, so now we can talk about some of the cytokines involved. Now, cytokines are going to be a bit confusing because within different contexts, they're going to have very different functions. So I'm going to sort of talk about a few of them within the context of allergic diseases and allergy immunology. But keep in mind that these could be very different depending on what realm of immunology you're thinking about. But really, the ones I want to talk about are IL-4, IL-13, and IL-9. And I might have a little bit on IL-5 in another lecture because it's quite important as well to eosinophils. But let's start with these three. So you can see over here that IL-4 is going to be a really big mediator of IgE-related hypersensitivity reactions because it induces IgE. Now we're fortunate that things such as interferon gamma and IL-21 can shut down this IgE isotype switching. So again, there's a balance between the cytokines that are in our body. So let's get into some of them a little bit more in depth. So the text I was reading described IL-4 as the prototypical allergic cytokine. I think that's pretty interesting. So the reason they say that is because it's so easily identified in pretty much any tissue that's undergoing an allergic inflammatory response. So it's quite ubiquitous in allergic inflammation pathways. It has a lot of effects. All of these are going to sort of increase inflammation. So we have things like increasing, antigen presentation with B cells, stimulating IgE receptor expression on mast cells, inducing adhesion molecules, steroid resistance, halting apoptosis, stimulating mucin production. So wow, a ton of different things that are sort of directly involved in how allergic inflammation occurs. And a lot of it can be traced back to this cytokine. So I want to talk about the receptors a little bit. So this is something that happens in the IL-1 family as well. But basically, the IL-4 receptors are a heterodimer, and they share a common chain with IL-13 receptors. Now, as you can see in this image over here, type 1 IL-4 receptors are only going to bind IL-4, but type 2 could bind both IL-4 and IL-13. So this is an interesting way to think about how IL-4 and IL-13 could be very, very related. Part of the reason why they have such similar functions and are so closely related in the context of allergic diseases is that they actually, kind of share a receptor, which is very interesting to think about. This is what I was talking about with IL-1, is that the IL-13RA receptors are sort of decoy receptors, and they can be used to suppress immune responses by sequestering IL-13, which is also really fascinating. So IL-13, these are pretty similar to IL-4, as I said, because they share that same receptor, but these are more longer distance. You can think more hormonal functions, whereas IL-4 is more, directly in the vicinity, so a paracrine function. And a lot of similar functions to IL-4. So we see mucus production, airway responsiveness, airway metaplasia, things like that. And interesting, if you've ever heard of Dupixent or dupilumab, this is a really popular biologic treatment that's coming out. It's a monoclonal antibody, and it's an anti-IL-13 antibody. And it's been pretty successful in asthma therapies. It's also used in eczema as well. It's very interesting to think of how, we can go from this sort of inflammatory molecule and connect it directly to its pretty popular thing. Now, IL-9, this is going to come primarily from Th2 cells, so that's another way in which they're directly related. And this is called the mast cell growth factor, sort of colloquially. So this stimulates production of mast cell proteases, which, if you recall, include tryptase, chymase, and also sort of primes these mast cells to respond to allergens by upregulating the Fc epsilon R1 receptor, which is the high affinity IgE receptor. And so, IL-5RA on eosinophils is also going to sort of be responsive to IL-9, and this is one of the ways in which we can sort of connect mast cells and eosinophils on a direct axis. So the cytokine that's this mast cell growth factor is also directly responsible for eosinophil proliferation as well. And just talking about eosinophils a little bit, IL-5 is really going to be the cytokine that we're always going to be connecting to eosinophils. So we can think of IL-5, they described it as the most important eosinophilopoietin. Basically, it's going to be really driving eosinophil survival, causing mucosal eosinophilia, producing chemotactic agents, causing accumulation of eosinophils into the tissue. So again, this is something we'll get into a little bit more in the context of specifically eosinophils and eosinophilia, but IL-5, really the connection there is to think about eosinophils. Another thing that I want to talk about is GM-CSF. So it stands for granulocyte macrophage colony stimulating factor. So this is going to be another cytokine which activates eosinophils and causes them to sort of increase their degranulation abilities, increase their responsiveness to chemotactic agents. So this is a really common trend in sort of allergic inflammation, is that a lot of these sort of cytokines might not have a direct function, but their function is to sort of, or a direct inflammatory function, should say, but their function is to really take these cells and prime them so that they're way more active or they're overactive in response to typical stimuli. So I'm going to skip this because I don't think it's too important, but you can read here that there are some other cytokines released by Th2 cells that are very involved in allergic inflammation as well. So now this is important. Let's return to that sort of Th1, Th2 balance and talk about how we actually get and handle the difference between these two. So I want to introduce the idea of this cytokine milieu, and basically this describes how when you have a naive T cell, the cytokines that surround it can cause it to start generally or just slightly polarizing towards a certain direction. And the way you get this cytokine milieu is really through a JAK-STAT, pathways that we talked about that lead to the appearance of specific transcription factors, which are going to start producing early amounts of these, archetypal cytokines, and these cytokines are going to create that milieu, which then helped to differentiate the T cell. So let's talk about some of the cytokines that are related to Th2 differentiation. So Th2 differentiation is sort of driven by IL-4, which is not surprising, as well as IL-19 and IL-25. So IL-25 is produced by epithelial brush cells, and it helps to induce IgE secretion. So again, very related to Th2 cells. But you also have TSLP, which stands for thymic stromal lymphopoietin. So this is a sort of cytokine that's going to promote the secretion of other Th2 cytokines, and it produces a lot of Th2 attracting chemokines as well. So this is a mechanism by which Th2 cells can recruit other Th2 cells. I also want to talk about some of the anti-inflammatory mechanisms in allergy, because again, we've talked so much about inflammation, but how do we downregulate inflammation, right? So again, like many things, it's a balance. So we do have things that are causing inflammation, but we also have things in our body that are capable of suppressing it. And we want to think about, in allergic individuals specifically, both sides of this sort of seesaw, and also why it's out of balance. Now, we might not think of cortisol as being an anti-inflammatory hormone in our body, but in the context of asthma, it just might be. So something we notice that's really fascinating is that when you look at nocturnal asthma, so patients that are having worsened asthma during the nights, this tends to correlate with the fact that during nighttime, we have less cortisol, because our cortisol, which is a sort of waking and stress hormone, is going to dip in the middle of the night where melatonin is the highest. So it's interesting to think that the stress hormone is actually potentially having some anti-inflammatory effects as well. This is supported by the fact that when we administer metirapone, which blocks cortisol release, we see that you tend to have increased late responses to allergens. So worsened allergy response when we block cortisol. So going back, we're always going to want to wonder, why is it out of balance in an allergic individual? Um, and a suggestion is this enzyme 11B hydroxysteroid dehydrogenase 2. So this is an enzyme in our airways, and it converts inactive cortisone to cortisol. So if you have more of this, you will have more cortisol. And so potentially this enzyme might be faulty in some ways in asthmatic airways, which is why nocturnal asthma is such a big issue. So interesting to think about. There are also various inhibitory cytokines. So I won't really go too in deep to most of these, but I do want to cover IL-10 because this one's important. So IL-10 is a very strong anti-inflammatory cytokine, and we see that it's markedly reduced in people with asthma, and it does things like inhibit antigen presentation, especially in this case allergen presentation, and it's produced by certain T regulatory cells and macrophages as well. So very interesting to think that we have this mechanism for downregulating inflammation, but it's not entirely present. And then finally, just thinking about some lipid anti-inflammatory mediators. So we talked about inflammatory mediators that were lipids, but now we can think about anti-inflammatory ones such as prostaglandin E2. So this one, in contrast to prostaglandin D2, this one inhibits macrophages in various cells. And if you were to administer this, you would actually see that it inhibits, airway responses that are caused by allergens. So it's very interesting to think about that as well. And then you also have these two molecules like poxins and resolvins, and these are both, just general anti-inflammatory ones. This one's generated from arachidonic acid, and it shuts down cystinolecgotrienes. And resolvins are one of the reasons why we might want some, omega-3 fatty acids in our diet. So that is it for this presentation. I know it's a lot, but I hope that it is at least interesting to think about. And really, again, the goal here is not to memorize everything, but to develop familiarity with these concepts. And I promise the later lectures, especially when we get to diseases, are going to be probably a lot more interesting to the average person, and also a lot more understandable now that we have the language to deal with them.